Yes, thanks for invitation and organizing great workshop. So you see the title of my talk. I'm interested in data integration, which is a great segue from Katya's talk, I think. So I want to acknowledge the work. This is joint work with lots of great people. John Fincy and Mingwei Tang are my former PhD students who finished their PhD when I was still at the University of Washington. Now John is at NIH and Mingwei is at Facebook, and also collaborator John Wakefield, Trevor Bedford, and Gitas Dudas on the couple of papers about this work. So I will start with a motivating example of that I'm sure most of you are familiar with. It's Ebola in West Africa, and I want to just illustrate what ideally we would like to do with this data, I think, is to look at the surveillance data, kind of the traditional surveillance data, confirmed probable cases. Ideally, as we learned during this pandemic, we also would like to know not just the positive tests, but negative tests that have been performed. But that's, at least for me, it's a more recent discovery how important this is to take into account. But normally you can download cases and you can pretend that it's not important to know how many people have been tested and how that varies across time. And then you also have genetic data. And from that, we can reconstruct the genealogy. And this is just a genealogy, a big kind of genealogy that Gitas Dudas with co-authors reconstructed in this very nice Nature paper that they published about kind of phylogeography and phylodynamics about Ebola outbreak in West Africa. So ideally, I think we would like to analyze this data jointly and maybe add more data, hospitalization data, critical care usage, mortality, just throw in all of that. And I would argue that's probably very important because we know that these models are hardly easy to estimate that we're trying to fit to these data right now. So again, this is just a background slide to set notation, or you by this time certainly familiar with all of this. There are multiple ways to view these SIR type of models. I prefer the graph on the bottom, which is a proper transition graph of a Markov chain rather than a strange S goes to I goes to R diagram that I never understand actually. This one I can understand. There's a state space that consists of triplets of counts, S, I, and R, and you can increase I by 1 by simultaneously decreasing S by 1. That's an infection. Or you can have a removal event which increases R by 1 and decreases I by 1. So that's a kind of a formal mathematical description of a model. You can view it at individual level, and it's, if you want to do sort of like, you know, something that people would probably call agent-based modeling, and you can actually think about individual people being infected. But more often we collapse these individual-based view into counts, and it's kind of population-based view, a collapsed Markov chain that actually is depicted on top. And so at the bottom, I'm showing you two representations, right? One is the differential equation representation on the left. On the right is the 3D realization of a stochastic model with the same parameters, the same population size. And you can see that even, you know, in the population of 10,000, the dynamics is not purely deterministic. It actually is not purely deterministic, even if you increase it to 100,000. And it's actually quite kind of a question for statistical inference when it actually becomes deterministic to the point where you can ignore completely all this stochasticity. Aaron King, who I believe is in the audience right now, at least he was a couple of minutes ago, has a paper about Ebola outbreak called Avoidable Mistakes something. And one of the avoidable mistakes is fitting ODE models to cumulative counts, which is he demonstrated quite nicely. It's a no-no. So so I'm interested in stochastic models. Deterministic models are fine as well. In fact, when I was asked to help the local healthcare agency with the COVID modeling, I also used deterministic models just because I panicked that I wouldn't be able to do everything in time to fit a stochastic model. But I would like ideally to be able to easily fit stochastic models to noisy surveillance data, which includes genetics data. And so when you start doing that, you will quickly find yourself in the space of hidden Markov models where X is your latent states, which is something you never observe, is your number of susceptibles, infectious, removed individuals, exposed, hospitalized if you have complicate compartmental models and so on. And then if you're dealing just with case counts, for example, with incidence, you can formulate X so that it actually tracks the cumulative incidence rather than prevalence, so you don't mistake prevalence for incidence. There are tricks for that. It's possible. And then what you actually observe is a noisy realization of a latent incidence, right? So every week you have some actual number of cases that have appeared in the population. What we observe is typically just a fraction of people who maybe were symptomatic and decided to get tested, and so on. And so what we're interested is in this posterior distribution of all these parameters that govern these models, are not, removal rate, and so on, giving these, for example, this vector of counts in the simplest possible case. And if you look at this, the likelihood is intractable because it's a likelihood of hidden Markov model where number of states is so large that you cannot do your usual matrix algebra and Baum-Walsh algorithm and all of that. And so you have to do something else. And something else usually means something like we've seen particle filters. That's probably the most versatile approach at the moment if you want to fit these models to data. And it's certainly being used in the MLE framework or in the Bayesian framework. What we're proposing, and actually it's not really, we are extending the work of Paul and Fernhat, who with colleagues proposed to use a linear noise approximation, which is a really very old technique from 70s about how to approximate these population processes with locally Gaussian processes. So what this, I'm not gonna dive into this old beautiful theory, but it basically replaces this intractable transition densities that cannot, or probabilities if you work in discrete space, or densities if you want to work with continuous representation, kind of stochastic differential representation of the SIR type models. It replaces them with locally with the Gaussian, multivariate Gaussian density. And so, which is really nice because now it becomes suddenly really a Gaussian latent state space model. It's complicated. It's not really, it's non-linear state space model. So the Kalman filter and things like that do not really work the same way you expect them to work here. But it's certainly possible to adopt modern MCMC. You can unleash Hamiltonian Monte Carlo or elliptical slice sampling that you use here, or lots of other techniques, because suddenly now you are in a tractable situation where your complete data likelihood, if you augment your likelihood with its unobserved latent trajectories, now decomposes into this product of tractable Gaussian transition densities and some maybe non-Gaussian likelihood. So this will be, maybe if you work with case counts, maybe this will be your negative binomial product of negative binomial probability mass functions. In coalescent, structured coalescent, this will be some sort of structured coalescent likelihood, and as we will see in a couple of minutes, and so on. But that's nice because that's these Gaussian, latent Gaussian models with non-Gaussian likelihoods. It's a bread and butter of computational statistics. It's lots of techniques are available to tackle that problem. How I spoke only about the incidence data so far, or case counts more generally. Adding genealogical data, again, there are lots of choices, and Katya talked about some of them, and one of them is structured coalescent. You can use birth death processes. We're gonna here, the approach we're taking really is kind of agnostic to what route you take, but here we're gonna use the Eric Voltz's structured coalescent approximation approach. There are other structured coalescent approximations that are also available, or birth death process available. And under this framework, we can write down the density of a genealogy, and we assume the genealogy is sampled under the sort of heterochronous assumption that structured sampling of sequences. This is kind of the very, very general density of the structured coalescent, where lambda is your coalescent intensity. And that coalescent intensity under simple Kingsman coalescent, this is just one, proportional to, or inversely proportional to effective population size. And under Eric Voltz's structured coalescent approximation, it's proportional to something else, which depends on SI in infection rate beta. So again, those are latent properties. If S is not changing much, as you can see, it basically kind of collapses to the Kinsman coalescent because S is a constant. And so in this case, your prevalence is basically proportional to effective population size, which is probably for Ebola. We're not going to take this assumption, but you will see that for Ebola, it's probably not a bad assumption. So you could fit Kinsman coalescent. The interpretation would be more difficult, but it's effectively Kinsman coalescent for Ebola application. So what people have done before, and most of the authors of these papers are in the audience, so at least some of the co-authors of the space of the papers. So you can take the ODE-based structured coalescent approach, and that's what Katya did in the previous talk. And it's a very nice series of papers starting from 2009. The most recent kind of methodological one is Volt and Sidroni, which is the BEAST2 module description of how to do for these very general, with a very general language of how to specify these models. The Bayesian inference via particle MCMC actually started with the paper by David Rasmussen, Katja Koelle, and Olli Ratman back in 2011. And actually, that's effectively what we're trying to do in a sense. We're trying to sort of do this paper, but without particle MCMC. You can think about it this way. Like, is it possible to do this without particle MCMC? And that's what we are trying to do. And these maximum likelihood iterated filtering, the approach that Aaron King at Ioannidis and co-authors have championed as well. So what we're doing, we're turning the Bayesian crank, the same way you can turn the MLE crank. The Bayesian crank here is we're trying to estimate posterior density of all model parameters and unobserved latent trajectories, like number of susceptible individuals at each time point, number of infectious individuals at each time point, or whatever your compartmental model specifies, how it specifies compartments. And it's proportional to its product of two likelihoods. There will be one incidence likelihood, for example, if you're working with incidence data, and this probably will be negative binomial, product of negative binomials, because that's kind of standard to account for dispersion in case counts using negative binomial distribution. This will be structured coalescent the way I showed before. It's tractable under the approximation, Quotes approximation or some other approximations. This is epidemic model, and it's important. That's what allows us to avoid using particle filters. This is epidemic model trajectory density, which is just a simple product of multivariate Gaussian densities that are derived from this LNA approximation. So that actually what makes the whole thing work, because ordinarily this is intractable. Even if you don't want to sum over X, you still don't know what those transition densities or probabilities are for large populations for sure, and complicated compartmental models. But here we do, and some priors. And so we turned Bayesian crank, and we're trying to infer all these parameters and latent trajectories jointly from sequence data, from genealogy data, even though genealogy, of course, not data, and we really want to integrate over genealogies as well, but we're not there yet at all. Even this is not kind of working the way I want it to work, to be honest. So we're usually trying these experiments with assuming incorrectly the genealogy is fixed and known, and then trying to relax it if it works. So let me illustrate the example that I started with. I'm going to use only one country. Why I'm going to use one country? Because we cannot do three countries. I would really like to do three countries jointly and account for importation, exportation events, and jointly analyze the cases and the sequences from all three countries. I cannot do it right now. It's too hard. So we're going to do one country. It has about 1,000 sequences from Sierra Leone. We're going to take one beast tree estimated somehow from beast. I mean, we know how, but supposedly it's not a better representation of a genealogy. Again, incorrectly assuming now this is data. We're going to take the case counts as well, the incidence data from WHO. We're going to fit an inhomogeneous SIR, where we will try to it's very simple SIR, except we are trying to estimate non-parametrically basic changes in basic reproductive number. So we're going to assume some smoothing, Gaussian marker random field smoothing, or autoregressive smoothing, and estimate the changes in R naught on a grid without really specifying the functional form of how R naught changes or change points or anything like that. And we're going to try to do it from both genealogies and the observed sequence data. Any questions so far? I'm kind of going a little quicker than I usually do, but Alexander, am I doing on time okay? I didn't notice when we started.
Yeah, you still have a couple minutes.
Okay, yeah. Okay, so let's look what happens in terms of results. So that's my last slide. So this is our estimation of this time varying R naught. And so it's a busy kind of slide. There are 3 colors because I want to really show you what happens if you use just genealogy, so just structured coalescence, just incidence, so just genealogy is in green, just incidence is in blue, and if you use join modeling, which is red. So one obvious thing that you notice, and that was noticed by, of course, the David Rasmussen and Katja Koele and Olli Ratman in the 2011 paper, you have sharper inference. Of course, if you had more data, you should have tighter credible intervals, and that's what these credible bands illustrate. Those are point-wise really credible intervals, but we're connecting them so they appear as bands. And so you have a tighter estimation of the changes in the basic reproductive number, which starts at about 1.5 and drops below 1 eventually. The uncertainty is much larger if you just use structured coalescent, and it's still larger still if you just use the incidence. As you can see, I would ignore the bumps in the density. We have all sorts of problems with MCMC convergence as preliminary results. It's not, we should run it longer to avoid all these kinks, but we do believe that the signal is real, is in effect, in sense that if you just use genealogy, you're basically recovering your prior in terms of the recovery rates or mean infectious period. So it appears, at least anecdotally, that just from genealogy and structured coalescent, it's not possible really to identify the infection rate. Sorry, not infection rate, the recovery rate. Or at least it's very sensitive to the prior, that's for sure. The incidence data does not agree with the prior and pushes it away from the prior, and the red is somewhere in between, which is the joint inference. I also want to point out the reporting rate. The opposite is actually true for the reporting rate in some set, not the opposite, but reporting probability, reporting of probability of seeing a case. That parameter is present only in the incidence model. That's the emission distribution of the incidence model. And typically you actually have a hard time recovering it because you can either, you can play with the parameters, and so reporting increases, you can increase latent prevalence and decrease the reporting rate. Only kind of the product of latent prevalence and reporting probability are really identifiable. So typically you do have hard time, even though here it doesn't recover the prior, the posterior of the just incidence data, but it's quite influenced by the prior. And so the joint inference actually makes the picture a little bit clearer, and it does show that the reporting probability is not as high as incidence data would suggest, or at least that's what genealogy is pointing to. And this is just kind of a posterior predictive sort of diagnostics where the joint and incidence only models kind of agree in terms of producing posterior predictive distributions of the data that we observe. So I think this is promising. To me, what's most exciting about this is not the computational aspects of it, probably, although that's what made it possible, but actually the thinking about how these combining the multiple data streams, how it really resolves all these identifiability problems that we typically have with these models. These, you know, we are not really, when we publish papers and we give talks, we're not always very upfront about how really ill-posed these problems are. They're horrible. They're not easy to fit. If you want to fit like a 20 compartment model to incidence data, I really want to see the prior versus posterior plots everywhere for every single parameter, because I don't believe all of them will have prior and posterior kind of disagreeing with each other, showing that you have gained some information from the data. So these data integration approaches, I think, are really promising. I think we should be doing more of that, but it's kind of hard. I will finish with references, acknowledging the very nice work on which we're building, you know, starting from Eric Bolt's and co-author's paper in 2009, and then this paper plus combined 2011 paper that I mentioned multiple times, Paul Fernand's LNA, introduction of LNA into inference for infectious disease models. And the work that I talked about actually is not published, but if you put together these last 2 papers, so the first paper by John Fincy, is the first author, is about LNA applied to noisy incidence data, and Mingwei worked on genealogies, and then Mingwei actually extended it to the joint model, but we haven't published that work yet. So, but if you put the last two papers together, that's kind of the talk that I gave today.
Thank you. So we started 1 minute late, so I'll sneak in a quick question from David. I really like the idea of LNA, but I'm wondering what's the worst that can happen if the density of the latent variables is strongly non-Gaussian and possibly multimodal. For example, epidemic without R not close to 1 that doesn't take off most of the time, but sometimes explodes.
Yeah, it's a great question. So LNA is not without faults, of course. It is an approximation. Moreover, it's an asymptotic approximation when population size is large enough. So actually, we are responding to reviewers right now with this John Fincy paper where we tried to break LNA because reviewers ask us to break it. Like when it breaks, when the time interval becomes too large or population is too small. And to be honest, we have hard time breaking it so far. It does. I mean, I'm sure it breaks eventually, but we applied it to 3 different regimes, kind of Ebola, flu, and SARS-CoV-2 regimes, hoping that one of them will break it. That didn't quite happen. But it does have hard time, for example, when you have one of the compartments becomes too low. So presumably that will be important. So if you have very low prevalence, for example, eventually we'll break it, I believe. And then we can see it, but only if you include the tail of the epidemic, for example. The very tail, if you start including really low prevalence, part of the epidemic, we can break it. But it's surprisingly robust. I was actually thinking it would break faster, but it's not breaking as fast as I even would like it, because for demonstration purposes, I kind of wanted to break it faster.
Thanks for the talk. We should move to the next speaker, and